of Oxford University Synthetic Biology Society, or SynBio.Oxford. We are glad this is the first talk of the Michaelmas term 2020, and today this will be delivered by Dr. Sam Rodriques, who is currently in Cambridge, Massachusetts, but he is opening the lab in Francis Crick Institute in London. So just a bit of housekeeping. I will first introduce the speaker, then Sam would deliver his pretty exciting talk, I think, which has multiple parts, and then after that, which will take, I think, around 30 to 40 minutes, you are more than welcome to ask questions to Sam. Please, for asking questions, do write them in the chat, and we will then just call on you to ask your question. We do this just to have some kind of housekeeping and ordering of the people. So the earlier you come with the question, the earlier you get to ask. So this is first of the couple of talks that we will have that we have scheduled in this term. So we have a talk with Sam today, where he'll be talking about the new tools for interrogating and manipulating biological systems. But next week we have a talk by Dr. Lucia Marucci from University of Bristol on modeling and controlling complex dynamics in mammalian cells. And then we will have a talk by Professor Jim Collins from the MIT, where he will talk about the topic that is soon to be announced. So please do tune in to all these talks, and please, follow us on Facebook, Twitter, and stay updated. And I'd just like to mention that Synthetic Biology Society at Oxford is sponsored by IDT, Integrated DNA Technologies, and we are really grateful for their sponsorship to them. This is how we can keep the large Zoom rooms so that more people can jump in. And yeah, so this is the basic housekeeping. And now today on Dr. Rodriques, who will be talking today. So Sam Rodriques is entrepreneur, technologist, and inventor in biotechnology space. He's invented a new nanofabrication method, a new approach to sensing neural activity with probes in the bloodstream, and new ways to extract spatial and temporal information from RNA sequencing. And he's now starting a laboratory focusing on translational biotechnologies at the Francis Crick Institute in London in the fall of 2020. And prior to that, he was an entrepreneur in residence at Battery, a biotech accelerator in Boston, Massachusetts. And he co-founded Saturn V, a company developing patient-oriented neurosurgical tools rather than the rocket that got astronauts to the moon, I suppose. And in spring 2019, he graduated with a PhD in physics from the Massachusetts Institute of Technology. And before, he graduated with MPhil in engineering at University of Cambridge, and with BSc in physics at Haverford College in Haverford. And he received numerous national awards and fellowships, including Hertz Foundation Graduate Fellowship, NSF Graduate Research Fellowship, and a Churchill Scholarship. So with that said, Sam, thank you very much for joining us today, and the floor is yours. Please feel free to share the screen.
Yeah, great. Thank you guys so much for inviting me, and thanks to everyone in the audience for coming. I'm looking forward to telling you guys about it. So that was a great introduction. I will, reiterate a little bit of it and just kind of give add a little bit more color to that wonderful introduction that Miroslav just gave. I am technically a theoretical physicist, or I'm a theoretical physicist by training, at least. So I started out in undergrad doing work in quantum information theory, and basically came to feel like there were not very many unsolved problems left in physics. Like if you look around at the natural world, like any physical phenomenon that you can observe in the natural world, you can basically explain down to the level of subatomic particles, right? Whereas, in biology and neuroscience, you look at the mouse and you're like, well, how does the mouse work? And the answer is, I have no idea. And so I ended up deciding for my PhD to move into biology, kind of quickly realized that the difficulty in biology is it's not a lack of theories, right? But rather a lack of data, and more specifically, a lack of technologies that will allow us to get the data that we need in order to understand the system. So I did my PhD with Ed Boyden, developing tools for neuroscience. And I'm an inventor at heart, so over the course of my PhD, I invented a bunch of different technologies, some of which Miroslav mentioned, things like spatial RNA sequencing, temporal RNA sequencing, 3D nanofabrication, some brain mapping stuff, some viral vectors, kind of all over the place. And the overarching theme, I guess, from my PhD was, or my overarching takeaway was that I got very frustrated with the difficulty of getting those technologies out of academia, right? So when you invent a technology in academia, you publish a paper, and the paper comes out again, hooray, it's a big deal. And then usually nothing happens, right? And I was sitting there saying, these technologies, they're so, they have so much potential, right? And yet we're not having impact because you publish the paper and then you move on to doing something else, and it doesn't, it never gets out into the world. And so I decided to found my own lab, which I'm, as Miroslav mentioned, I'm currently in Cambridge, Massachusetts, which is where MIT is, which is where I did my PhD. But in December or January, I'm coming over to London to start my lab, the Applied Biotech Lab at the Crick Institute. And the thing that my lab is basically that I wanted to focus on in my lab is being more intentional about not just developing technologies or being intentional about developing technologies that will be revolutionary for biology and medicine, but then also being intentional about translating those technologies out into the world and actually achieving real impact with them. Okay, and so that's what I'm going to tell you guys about. I have to say at the beginning that I have fully funded PhD and postdoctoral fellowships available. Their PhD ads is online right now at the Crick. And my goal is to produce trainees, to produce postdocs and PhDs who, like I said, know both how to develop the technologies, who are both extraordinary world-class technologists, but who also know how understand the path that the technology has to go through to achieve impact, know how to deploy the technology, right? So I would say there are 4 things that I want the people in my lab to do. It would be to learn how to identify major unmet needs in biology and medicine. So this is to say, learn how to distinguish between problems that people have from problems that people don't have, which are that's a really, really difficult thing to figure out, that the problem that you're solving is actually a real unmet need. To then build technologies to address those unmet needs. Obviously, we're an academic lab, so then go and publish papers in Science and Nature, and then finally spin the technologies out into startups or get them adopted into major multinationals. Basically, know how to get the technologies across that barrier. So if you're interested, please, you can go to my website, appliedbiotechlab.com. You can email me, sgr@crick.ac.uk. I would love to hear from you. And also, this talk will be maybe a little bit different because I'm first going to talk a little bit at the high level about the vision for the lab and the structure of the lab, and then afterwards I will talk some about science and some of the projects that we're going to be working on. Okay, so the basic problem that I'm talking about is the problem that the academic incentive structure today values novelty rather than usability, right? And so if you go and you talk to academics who are working on technology development, you hear them say things like, if I commercialize this technology, the company will compete with me, right? Or, if we help other people use this technology, we're going to lose our advantage. Or the one that really, kind of drives me nuts is the goal is to build technologies that are so important, other people have no choice but to use them. Which all of these basically, de-emphasize the role of the academic in actually getting the technology out the door. right? And in fact, that the incentive structure is such that often what academics try to do is to develop technologies that are actually hard to use, right? Because then they kind of create a niche for themselves. And basically what I'm talking about is that I think that the way to have the most impact is to develop technologies that are easy to use, that address real problems, right? And so this is what I refer to as need-oriented biotechnology, right? Technologies that are easy to use, technologies that can be spun out, can be deployed. And basically just saying we're gonna build technology, we're gonna get it out, then we're gonna go and invent something new, right? And so the 4 areas that I expect my lab to work on at the beginning are here. Basically, brain mapping or connectomics, kind of something like the genome project for the brain, viral vectors for gene therapy, liquid biopsy, so being able to identify diseases, cancer or neurodegenerative disease, things like that, from blood samples, and spatial transcriptomics, so being able to identify the spatial structure of gene expression. And these are all areas where if you manage to invent a new technology in these areas, it can be really transformational for biology and medicine. But there needs to be some upfront work to be done on the technology development. And so the way that I'm structuring my lab a little bit differently in order to, the philosophy is a little bit different in order to kind of focus on building deployment into our thinking from the very beginning. And the ways in which we're different was basically 3 ways. First are in the kind of people that we're surrounding ourselves with and the kind of people that we're hiring. Secondly is in the problems that we choose to tackle and the way in which we identify those problems. And the third is the actual process that we use to come up with solutions to those problems. So on the side of people, I think one of the major things is I really believe strongly that if you're building technologies for deployment, you have to talk to people who are good at deployment. We're building a network and an advisory board of entrepreneurs, executives, philanthropists, investors, people who know, who can evaluate technologies and help you figure out what the correct path is, right? And so some of the people who are going to be on my, who have agreed to be on my advisory board are people like George Church, who obviously is one of the pioneers of genomics at the Wyss Institute, and who's known, as one of the most entrepreneurial professors in the world. Annalisa Jenkins was the former executive vice president, global head of R&D at Merck Serono, which is one of the largest pharmaceutical companies in the world. So she really understands what pharmaceutical companies look like from the inside, can really tell you whether, if you're working on technology, is it something that the pharma companies can use, right? Sam Lapidus, who is the founder and CEO first of SciTech Corporation, which and was acquired for $6.7 billion. And then was the founder and CEO of Exact Sciences, which is the maker of Cologuard, which is a non-invasive colonoscopy, like colon cancer screening platform, which IPO'd and now has a $14 billion market cap. So he's one of the most successful entrepreneurs in the world in the diagnostic space, right? Again, you just can talk to him and be like, hey, we're thinking of this kind of a diagnostic. Is this like a does this have potential, right? And you just get feedback from these people. I mean, the first thing is you get feedback from these people. Second of all, if these kinds of people are on board, also Jason Malad is I left him one of the incubators associated with the University of Cambridge. And the point is that if you have these people on board, not only can they give you feedback and help you figure out what the right path is for the technologies and how to get the technologies out, they can also, having them on board is a way to, it gives you leverage, right? When you then go and you talk to investors and you're like, Annalisa Jenkins and Sam Lapidus are on board with this, then, that's like a huge positive signal for the investors. When you go and you talk to the technology transfer people and you're like, hey, we have these investors behind us, right? Again, it gives you leverage. So that's the, on the advisory board, and also we're forming partnerships. So for example, Petrie Biotech Incubator in Boston will be one of our partners, and they, the advantage there, people who are in my lab will be able to go to their like kind of breakout, which is their course automatically get admitted to their course to try to, where they basically teach people about what the path is, for kind of deep tech startups and things like that. I want to emphasize, of course, like this is, the main thing that we're going to do is technology development. I'm going to tell you guys about some of the technologies that we're developing, but it's really important to have this kind of ecosystem around you so that you're able to have impact. The second is the way in which second way we're different is the way in which we identify problems. So when you choose, a problem area that you want to work on, basically just engaging the stakeholders, identifying the stakeholders and engaging them to figure out what the biggest unmet needs are in that problem area. This is something that I feel like really shouldn't have to be said, but most people in academia don't do this. Usually in academia, when you want to figure out what problem to work on, the way you do it is that you go and you read a review. But that only tells you what other academics think the problems are. And in reality, what you want to be doing is you want to say like, oh, who's going to be the user of this technology and what do they think? So if you're going to be developing a new viral vector, you've got to go to find the clinicians and say like, what are the problems, right, with the viral vectors right now? You got to go to the investors and say, what is investable, right? You got to go to industry and say, what can be manufactured, right? And figure out there like what the problems are from their perspective. And then once you have figured out what the problems are from their perspective, coming up with objective criteria that allow you to kill projects, right? So, another example is kill projects if they're not going to satisfy, if they're not going to be able to have impact, right? So another example is like, suppose you want to come up with a diagnostic for antibiotic resistance, right? Well, you got to understand, you could go and you could talk to the doctor. You could have some amazing diagnostic, right? But you have to go and you talk to the doctors, and the doctors will tell you like, oh, we would only be able to use it if it comes back within like 2 hours or something. Because if it takes 24 hours to come back, then we can't use it. We're just going to put them on broad spectrum antibiotics, right? And you want to know that from the beginning so that you don't spend all your time like working on a diagnostic. It could be a great diagnostic, but if it is, requires you to do Illumina sequencing, for example, and the Illumina sequencer takes 24 hours to run, like thanks, but no thanks. It's like it could be the best diagnostic in the world, but it's not going to get used, right? And so those are the kind of criteria that you need to know so you can kill projects. And I just really want to emphasize here, I think it's always extremely important to be killing projects. The goal always should be, whenever you're working on a project, the goal should be to prove that it's not going to work as quickly as possible. And that's also something I want to emphasize because, if people are looking at, for example, the PhD advertisement that I put out, I do not expect for the students or postdocs that come to my lab, I do not expect that the problem or the project that they come intending to work on will be the project that they end up graduating with or the project that they end up publishing on. I expect that the projects will fail. I expect that everyone who's in my lab will be working on projects that fail. And then you will move on and you will learn and you will then work on new projects that will succeed, right? And that if you're not failing, you're not learning. If you're not failing, you're not taking enough risk. I guess I started over the course of my PhD, I started 26 different projects, of which most of them lasted about 2 months. I would say was like the median length. And of those 26, 6 succeeded, 6 different papers. And 3 of them are still going. So, you just keep iterating and eventually you find stuff that's really, really cool. Okay, so basically that kind of summarizes what my vision for the lab, building technologies that, then if they're useful for industry can be licensed, if they're useful for scientists can be spun out into core facilities, if they represent market opportunities, and if the best way to get them into the hands of patients or users is via a company, starting a startup, but just building deployment into our thinking from the very beginning. So with that, I'm going to move on now to talking about some of these projects. But first, I will preface this by saying a few words about what we're not going to talk about. So I mentioned these 4 areas at the beginning, and unfortunately, viral vectors and liquid biopsy, these areas are so extremely hot, and so there's so much interest in them right now and the ideas that I have around them are not yet protected, and so I can't talk about them in a public forum like this. So if people are interested in those areas, please, by all means, reach out to me. I'd be happy to talk about them privately. But otherwise, it would invalidate the IP if I talk about my ideas there publicly right now. So that leaves, well, I'll tell you about a story about spatial transcriptomics. I'll tell you a story about brain mapping. And I also just want to mention before we dive in there that I've worked on a bunch of different stuff. I've everything from like recording neural activity through optical fibers placed in the vasculature to like a different method for single molecule protein sequencing, to like this 3D nanofabrication method that basically involves embedding materials inside of a hydrogel and then shrinking those materials down to shrinking that structure down to the nanoscale. So I'm not going to have time to talk about those today, but if you're interested in those kinds of, if any of that stuff speaks to you as well, please by all means reach out. Okay, so the first scientific story I'm going to tell you is about this technology called Slide-seq, which is a spatial transcriptomic technology. So Slide-seq came out in Science in March of 2019. And this was a project that I worked on with, I invented it along with Bob Sickles, who's in the top right there, who's a good friend of mine. And this was in the lab of Evan McCosco and Fei Chen, who are, Evan McCosco is in the top left, Fei is at the bottom, who are now both professors at the Broad Institute in Cambridge, Massachusetts, sorry, in Boston. And Fei and I, actually, the backstory here is that Fei and I, so my PhD was officially with Ed Boyden, who's at MIT. Fei and I were graduate students together in Ed's lab, and he was about 3 years ahead of me. So he graduated, he started a lab at the Broad Institute, and I kind of went with him to work on this project. And I think Ed Boyden deserves a shout out because he was very gracious. He had no part in this project, and he was very gracious in being willing to let us publish it, and without him being an author and just basically being willing to let me work on it. So it definitely deserves a shout out to him. But so to set the stage here, biological tissues are structured. They have spatial structure, and that spatial structure is also is structure in the patterns of gene expression in those tissues, right? So if we think, for example, about the brain, the cortex in the brain is divided into 6 layers in mammals, and those layers are distinguished by the kinds of cell types that are present in those layers and the density with which they're present, right? And those cell types are distinguished based on the genes that they express. And so really, those layers emerge from spatial patterns of gene expression, right? Likewise, in the case, like very famous case of development in the gene expression of the Drosophila embryo, right? Like you get these patterns of gene expression that lay the plan, the body plan for the organism, right? And then obviously, in the case of many different pathologies, right? Cancer, in this case, the picture here, amyloid beta, Alzheimer's disease, these local pathologies have local effects on gene expression. And so there's a big, we really want to be able to study, there's a lot that we can learn about these systems by looking at the spatial structure of gene expression, right? But at the moment, the most powerful tool that we have to look at gene expression discards spatial structure, right? And this is basically single cell RNA sequencing. So, in a single cell RNA sequencing experiment, what you do is that you take your tissue and you dissociate the tissue. So you break it up into its constituent cells. And then you can either, for example, in the diagram here, you can sort those cells, you can basically encapsulate those cells in droplets with microfluid, with like little beads, barcoded beads, which can then capture RNA from the cells, and that allows you to figure out which RNA came from which cell. This is like the drop-seq or in-drops, or this is what's behind 10x. Genomics is a single cell platform. You also can do like fact sorting of cells for single cell sequencing, but all these methods basically involve dissociating the cells before you sequence them, right? So you lose the spatial information in the process. And so if we think about what techniques there are for doing RNA sequencing with spatial resolution, they basically break into two categories. So on the one hand, there are the hybridization-based methods, right? Like on the left is MERFISH or StarMap, and on the left, down here. And those methods basically can provide you with really like single cell resolution, because you're actually using a microscope. You can actually see the cells, right? But on the other hand, you have to specify in advance exactly what genes you want to look at, right? So you have to go in with a hypothesis and know exactly what you're looking at. Those protocols are typically very hard to do. So you typically have to do a lot of work to adapt them to any individual tissue type. On the other hand, there are the sequencing-based spatial transcriptomic methods, such as this method from Stahl et al., published in Science 2016, which is now being sold by Connectomics as Visium. I mean, the original paper is just called spatial transcriptomics, but basically you can put tissue down onto an array, and you can get you can sequence the RNA that's present at every point on that tissue, and there you get genome-wide transcriptomic, like a genome-wide view of the transcriptome-wide sequencing information, right? But you don't get access to single cells. And so what we wanted to do here was come up with a method that bridges these two paradigms that would give you transcriptome-wide RNA sequencing information with single cell spatial resolution. And so to do that, we took inspiration from this drop-seq method and figured if we could only figure out a way to associate a spatial location with each of these beads, right, then we would be able to do what is essentially single cell RNA sequencing, but with spatial resolution. And so the way that we went about doing that was that we took the beads, they're basically exactly the same beads that you do in single cell, that you use in single cell RNA sequencing, and we deposited them onto a surface, onto like a lower glass cover slip, into these arrays. So these over here, these white things are 3 millimeter circular arrays of beads, and then and this is like a bit of a zoomed in picture, so you can see each of the white dots there on that in that image is its own bead, right? And if you zoom in on them, here they are. These are the beads. It's a monolayer of beads on the surface, and we refer to each of these arrays, by the way, as a puck, because the original, like the initial version, they look like hockey pucks, basically. And then once you have these arrays, what you do, so each of these beads has a barcode on it, right? And so you can go and you can actually sequence the barcode in situ, right? So we built this, we got this microscope, and you put the arrays onto the microscope, and you go through, and you're directly sequencing the array, sequencing the beads while they're on the array. And so this is an example of one round of sequencing. You see 4 different colors there for each of the different bases of DNA. And then on the right, you see the base calls, and you go through many different rounds of sequencing, about 14 rounds. And what you come up with is for each bead, both its location and its barcode. Okay, and the thing that's really important to emphasize here is that this step of the process, right, can be done in batch at the beginning, right? So you create a lot of these arrays all at the beginning, and then you basically cut them up to use them later. So once you're ready to use one, you take it out, you take freshly frozen tissue and you section that tissue onto the surface. The RNA transfers from the tissue onto the surface, and you do some very basic, wet lab protocol to get the RNA. Basically you do like reverse transcription, you do digestion, and what you end up with is these beads, a library of these barcoded beads, where every bead has on it a library that's very similar to the kind of library you would generate in single cell RNA sequencing experiment, right? And so then you can go and you can match the barcodes, right, that you get out of this sequencing with the barcodes that you get out of the Illumina sequencing in step 3 with the barcodes that you got out of your sequencing on this array in step 2. Okay, and so then once you sequence it, you have data that's essentially the same as single cell RNA sequencing, so you can do all the same stuff with that data that you would imagine doing with a single cell RNA sequencing data set. Like you can identify cell types, but then because each of those cell type or each of those beads is associated with a location in space, you can just put those beads back into spatial context, and you get what is essentially an image of your tissue, right? Where every pixel in that image is annotated with all 20,000 genes in the genome. So this is a coronal section of the mouse brain. You can see the hippocampus down here in orange and in pink. You see the cortex up here, and then you see the corpus callosum in blue. And it's really important to emphasize here that there was no microscope involved for like the end user, for the person who used the array. There was no microscope involved in making this image of the tissue. And just for people who are not familiar, who are not used to looking at the mouse brain, this is a DAPI image from like the next section of brain. So showing you where the cell nuclei are. And so you can see it's a very faithful, very faithfully preserves anatomy. And actually, the whole goal here was to get the single cell resolution. And so I can now show you that not only is it faithful at preserving anatomy, it's very faithful at preserving anatomy at the single cell level. So this is zoomed in here, is an image of, in blue, so in yellow, we have the habenule, which is part of the mouse brain. The habenule is separated from the ventricle by a single cell layer of ependymal cells, okay? The ventricle is here in green, the habenule is in yellow, and that blue line of beads is the ependymal cells, okay? So you can actually make out single cell features like that. And so that, a lot of what I'm going to talk about today is the brain, because, that was kind of our focus. But I just want to emphasize that this method can very easily be applied to lots of other tissues. We've applied it to, I think we have liver and kidney up here. We have applied it to many different parts of the brain. We applied it to human brain tissue. We've applied it to like regenerating axolotl limbs. We've applied it to a bunch of different kinds of cancer tissues. We applied it to, patient-derived xenografts, so like, patient-derived cancers that have been placed inside mice. So you can do all sorts of stuff, and it works very naively on lots and lots of different kinds of tissue. So I'm going to tell you about 3 things that you can think about doing with Slide-seq. The first thing that you can think about doing is using it to identify the location of cell types that you already know about. So if you have a single cell RNA sequencing library, we came up with a way essentially to map the Slide-seq beads onto the cell types that you know about from your single cell RNA sequencing library. And the way that you do that is basically by breaking down your single cell RNA sequencing library using, clustering it using non-negative matrix factorization, and then projecting the Slide-seq beads onto the clusters that you identify in the single cell RNA sequencing, and you do that within the NNF space. And what this allows you to do, there we go, is to identify even things that are even very rare cell types within your tissue. So this is a sagittal section of the mouse hippocampus, and you can see pretty common cell types here, such as the dentate gyrus, which is in blue, CA1, which is in green, but then you can also see much rarer cell types, like for example, these CA2 cells in pink, and I guess probably my favorite thing is the neurogenesis cluster. So neurogenesis occurs on the inner layer, there's a single cell layer of neurogenesis in mice on the inner side of the dentate gyrus, and you can make that out here in red. Right, and so you can do this on many, many different sections. So this is we did this for like 100 some sections, 130 sections or something. And you can build up what is essentially a 3D representation of the tissue right here. Like this is the 3D representation of the mouse hippocampus. And so this is powerful, and we've been able to use this to identify rare cell types in a bunch of different a bunch of different tissue types. The second application that I want to tell you about is basically identifying the effects of pathologies on local gene expression. Okay, so what this means is, like I kind of mentioned at the beginning, there are a lot of different pathologies, right? Like cancer, is a good example, or injury, right? That affect the local tissue, and in order to develop therapies for those or diagnostics for those diseases, you would like to be able to identify what the effects of those pathologies are on the surrounding tissue. And so, as a first example of this, basically we applied Slide-seq to study traumatic brain injury, and our model for traumatic brain injury was stereotactic injection. So we took a mouse, and basically we use a standard kind of viral injection protocol. So this is injecting, used all the time in neuroscience, basically to inject viruses into the brain to allow you to study, study different brain populations, for example, for optogenetics or things like that. And so we use that protocol, and then we measured with Slide-seq to see if we could visualize the formation of the glial scar in the days after injury, right? And if we could look at what are the effects of that injury on local gene expression. And I'll just kind of walk you through this. So in the top left is again the mouse brain, and you can see right here in in the top left is the mouse brain as visualized using Slide-seq. And then over here is a DAPI image, and you can see actually where the scar is in the brain. That's like that dense area of cells in DAPI. And then if we go in, we can, as I showed you before, you can identify all the different kinds of cell types that are present and where they are. So, and this basically lines up with what people had already known about glial scarring, which is that you can identify fibroblasts, which are kind of in the middle of the scar, followed by this astrocytic scar around the outside. Then these microglia, which are kind of all throughout the scar, and finally the neurons get pushed to the outside, right? But the thing that we were able to discover that was super cool using kind of assumption-free methods for identifying gene expression patterns in the Slide-seq data was that we were able to discover that in the neurons surrounding the scar, immediate early gene expression remains upregulated for weeks after the injury. Okay, so immediate early genes are genes that neurons express when they fire. They're involved in plasticity. And we found dramatic upregulation up to 1 millimeter or 2 in those neurons away from the scar, even 14 days after the injury. And this is super important because, like I said, this is a kind of protocol that neuroscientists use all the time when studying neuronal activity in animals. And so there's a danger here that actually using this protocol will disrupt the activity of the neurons, can chronically disrupt the activity of the neurons, right? Even after the scar is healed, the activity may remain disrupted. And those are exactly the neurons you're trying to study, right? So this can be a confounding effect, and a good example of how we can use Slide-seq to look at gene expression in the surrounding pathology. So the last application I'll tell you about before we wrap up with Slide-seq is spatially differential gene expression. So differential gene expression is a very common thing that people like to is something people commonly like to look at, where basically when you have 2 different cell types or 2 different conditions, what you do is you compare them. Often in a single cell RNA sequencing experiment, you'll compare them to figure out which genes are upregulated in one in one cell type, or upregulated a different cell type or a different condition, right? And our idea was that there may be cases in which you're not actually able to identify differential gene expression in a given population, but in which the differential gene expression may actually be spatial, right? So it may not be that there's like an overall difference in expression between 2 different cell types, but rather that there's some genes are expressed in one location and not in another location, right? And that patterns of expression like that you may not be able to pull out. You may not be able to pull them out using single cell RNA sequencing, but you may be able to pull them out using Slide-seq. And so, as an example, we turned to the cerebellum. The cerebellum is an interesting case for this because the cerebellum is at the back of your brain. It's involved in motor control. It's a very old part of the brain, and it's essentially as old as the vertebrate lineage. It kind of co-evolved with the spine. And it's pretty interesting because basically there are, there's this layer of cells on the outside of the cerebellar cortex which are called Purkinje neurons. Okay, and Purkinje neurons are commonly implicated in things like motor learning and things like that. And basically, before this study, there was only one known kind of Purkinje neuron, right? Like there were not known to be many subtypes of Purkinje neurons. If you do single-cell RNA sequencing, you would only get one cluster, right? But it was known from immunohistochemical studies that actually there is differential expression in those Purkinje neurons, right? So just so that people know what you're looking at, so this is a sagittal section of the cerebellum, and this layer of cells on the outside there, the Purkinje neurons, and you see PLCB3 is an example of one gene that's expressed in the Purkinje neurons, but only more posteriorly, whereas PLCB4 is also expressed in Purkinje neurons, but mostly anteriorly. And this pattern of gene expression is known as the zebrin staining pattern, because the first gene that was discovered to be expressed this way, if you stain for it on like a whole mount cerebellum, you get this like very nice zebra stripe pattern. And so what we wanted to ask was basically, can we use Slide-seq to identify systematically patterns of gene expression, or sorry, it says systematically which genes display this kind of zebrin staining pattern. Okay, and so it turns out that the answer is yes. So we went in and we did Slide-seq on a bunch of cerebellar sections, and we clustered the genes according to their spatial, their pattern of spatial expression. We got these two really nice clusters, right? One of which was clearly zebrin positive and one of which was clearly zebrin negative. And if you plot these in space, right, where like the zebrin positive, I guess, is teal here, and the zebrin negative is red, you get this wonderful separation of the cerebellum into two different compartments based on whether they're zebrin positive or zebrin negative. And in the process, we have now discovered, 30 or 40 new genes that show this gene expression pattern, right? But that's not all. We also were able to discover in the process, because we're using these kind of unbiased analysis methods, we were able to discover a bunch of new gene expression patterns. This is one of my favorite, which is this gene H2D1 is one of a number of examples that we found that are expressed in Purkinje neurons, but only outside of this one region of the cerebellum called the nodulus. So the nodulus is down here, right on the bottom. It's an older version of it's an older part of the cerebellum that's involved in vestibulo-ocular learning. So basically, like allowing your eyes to follow different targets. And you can see that this gene H2D1 is expressed everywhere except in the nodulus, whereas this HSPB1 gene in blue is a highly upregulated in the nodules, right? And so this is super interesting because on the one hand, H2D1 is an MHC class molecule, which are now known to be involved in neuronal plasticity, right? So this could be indicative of different forms of learning in the nodulus versus the rest of the cerebellum. On the other hand, HSPB1 is a heat shock protein, which may be involved in like the nodulus is known to be particularly resilient to disease, and that kind expression of genes like HSPB1 could be part of the reason, right? So there we discovered a bunch of other patterns as well, which I encourage you guys to go look at the paper if you're interested. But I can say that this past March, Evan McCosco and with an RA in Evan McCosco's lab named Carly Martin, who's amazing, published a preprint on bioRxiv, and the paper will hopefully be coming out soon, in which they actually went through more systematically and characterized using Slide-seq and also using single-cell RNA sequencing, and were able to identify all these clusters and find there are actually nine different kinds of Purkinje neurons. Okay, which is like a really just kind of like paradigm shifting discovery about Purkinje cell biology, and the roles of these different cell types is obviously not yet known and will be a major source of future future discovery. So what I want you guys to think about, basically, what I want you to take away from this is that whenever you're looking at a differential gene expression matrix, right, like this one here, you should think that there's also some kind of a map, like this, right? Like there's a spatial component to this differential gene expression as well, and Slide-seq allows you to get that out. So that was what we found in that in that paper that was published in March 2019. Since then, we've made a bunch of improvements, have improved the yield by about a factor of 10, so that the yield is now similar to DropSeq. And we're currently working on trying to figure out how to commercialize this technology and get it out so that people are able to use it, all across for many different applications. So I see that I'm running. I'm running. This is taking it's taking me longer to get through this than I thought. So I'm just going to run kind of quickly. Well, I don't know. Should we just do, Miroslav, what do you think? Should we just do questions, or what do you think?
Well, I think, well, so we got perhaps 10 to 12 minutes, but I think, yeah, perhaps just continue for a bit. If someone has any question, please feel free to post them in a chat. But until then, perhaps let's just go on with the talk.
Sounds good. So I'll just move on to the other thing that I want to tell you guys about was brain mapping. To lay the groundwork here, all psychiatric diseases are associated with changes in synaptic physiology, right? And so being able to map the brain, being able to actually map brain at the single synapse level is super important in order to understand psychiatric diseases, right? Like if you take a brain of someone with depression and you slice it, right, you're not going to see overt pathologies, right? The pathology is actually at the level of synapses. It's not like cells are dying, it's at the level of synapses. And so the problem that we have today is that the brain mapping technologies that we have are too slow and too expensive to be adopted widely. And so basically the method that is used is called electron microscopy. This is one example here of a single electron microscope image of the brain, where each of these little circles that you see is a different neuronal process. And basically what you do is you take sections, you take images like that and you stack them up until you make a beautiful, a big cube like what you see over here. And then you can reconstruct that into a cube that looks like this, where you actually now have the wiring diagram of the brain, and it's amazing and super exciting for neuroscience. But also, making a cube like this, which is scarcely 100 microns on each side, can take hundreds of thousands of person-hours to reconstruct the processes from these images, and also leaves out crucial information, like information about the proteins that are present at synapses, which are actually the things that drive the function of the circuit. Okay. And so where we are right now, there's an analogy to be made in terms of where we are right now with brain mapping and where we were with DNA sequencing, right, 20 years ago. So this is an image of the Whitehead's sequencing core in 2001, right? The Whitehead at that time was one of the leading centers for sequencing the human genome, right? And they had this whole room, right, dedicated to sequencing. And now you can just buy a NextSeq, which has 100,000 times higher capacity, right? And will just like sit on your bench. And so where we want to go is from the current state, which is on the left here, this is an image of the Allen Institute's brain mapping facility, which is a giant room. There's 6 electron microscopes in it, and these electron microscopes are so old they can't even buy like the PCBs that control them anymore, right? Something like that, in order to make these like electron microscopy connectomes over to something that would be like a benchtop device, in 10 years or 20 years, that will allow any neuroscientist in the world to be able to integrate connectomics into their study of the brain, right? And this is not just kind of an idle curiosity. This is becoming like a super, super hot area. So the Brain Initiative was an initiative that was launched by Obama, like, I don't know, 2015 or something, 2014, which basically was like a $5 billion project to develop tools for neuroscience. And they're about to renew that project and extend it. So Brain 2.0, and one of the priorities for Brain 2.0 is to map the entire mouse brain connectome, right? And so basically where we are right now is we're like at the same place where we were in 1990, where we're getting ready to start mapping, the first full connectome of a mammal. And we need to be thinking about what comes next. And so what I want to outline for you guys, and just pretty briefly, is the 3 components of a next generation connectomics technology that I'm developing. And those components are super resolution optical microscopy, multiplexing, and molecular barcodes. I'll go through those in turn. So the reason that everyone uses electron microscopy is because historically electron microscopes have been the only, electron microscopy has been the only microscopy method that gets sufficiently high resolution to actually visualize individual synapses, okay? But now, in the past 5 years, with the advent of new super resolution microscopy methods, like these are examples from expansion microscopy, we can now actually visualize synapses with optical microscopes, right? So that's on the right, what you're seeing is like a 5x expanded piece of brain tissue where the pink and teal are like a postsynaptic and presynaptic protein respectively, showing you where those synapses are, right? And the major advantage of optical microscopy here is that it's compatible with protein imaging. So you can do things like look at the proteins that are present at the synapses, and you can also use barcoding, right? So along the lines of looking at proteins, the second component is highly multiplexed RNA and protein imaging. So there have been a ton of techniques that have been invented in the past few years, allowing you to look at dozens or hundreds of proteins in the same sample, right? So you can really get a pretty holistic visualization of what kinds of genes are being expressed by each different cell. And finally, the third component is barcoding. And barcoding is really important, right? Because we want to be able to trace really long processes, but we don't want to have to trace like the entire axon. So we want, if like a neuron projects from the thalamus to the cortex, we want to be able to follow that projection, but we don't necessarily want to trace the entire white matter all the way through, right? And so barcoding is the idea that there may be ways using molecular imaging to identify neurons uniquely regardless of where they are in the brain. And so there are a lot of different ways of doing this. You can use RNA. So like each neuron can express a unique piece of RNA. Each neuron could express a unique, and so that's a method that a bunch of people are working on. We're taking a different approach, which is a protein barcoding method, where each neuron expresses a unique combination of proteins, right? And basically, it's based on Brainbow. In Brainbow, every neuron expresses, you have 3 different fluorescent proteins, and every neuron expresses a random subset of those 3 different proteins. So that's the image that you're looking at here. So each of the neurons ends up with a different color. Unfortunately, if you only have 3 fluorescent proteins, you only get 2 to the 3rd possibilities, which is 8, which is not enough to uniquely barcode all the neurons in the brain. But what we're working on is basically making a version of Brainbow leveraging this like protein multiplexing approach that would give you a total of so 30 different color channels. So it's 2 to the 30th. So it's about 1 billion different combinations, which is enough to uniquely label all the neurons in the mouse brain, right? And so this is basically what it looks like. You start out by delivering your barcode to the neuron. So you deliver your barcode to the mouse, for example. You then do expansion and label the epitopes that you want to look at. You then go in and image this barcode, right, by using this multiplex imaging, multiplex antibody staining techniques that we have. And then finally, you go in and you label endogenous, you image endogenous proteins like synaptic proteins, right, in order to be able to get synaptic phenotype, right, and in order to be able to actually see where the synapses are. And so, just for time reasons, I'm not going to run you through. Basically, the hard part here was coming up with this protein barcode, this multiplex Brainbow technique. We use these methods called spaghetti monster fluorescent proteins. I won't talk about that too much, but what we have right now is about 17 different channels. We have another 17 different, essentially fluorescent proteins that can be labeled uniquely with antibodies. We have another 36 that we're screening through, so we think we'll be able to get to around 20 to 30. This is a postdoc, Bobe, who I've been working with on it. This is just like preliminary data to give you an example of what this should look like when it's done, right? So on the right, you have a 4-channel Brainbow image in culture, right? So you can see the different neurons, and it's you can trace their processes based on the color. And then on the right, you have in the same sample images for 3 different proteins, 3 different synaptic proteins, NMDA receptor, which is a neurotransmitter receptor that's strongly implicated in learning, and Homer and Bassoon are 2 scaffolding proteins that can be used to identify synapses. And when you put it all together, what you get is an image that looks like this, where you can actually trace the neurons, because you have the different channels, you can trace the neurons throughout this image, but you can also visualize those puncta are showing you where the different synapses okay? And so we've been doing this, we've been scaling it up to do it in mouse brain tissue. We're doing it in iPSC-derived neuron culture for drug screening purposes. And we've also been doing it actually in human tissue from epileptic patients that you get from epileptic patients who had surgery. So this is just an example of our first experiments getting this to work in human tissue. And so I think that it's actually possible with this kind of a method that you could get it down to the point where you would be able to do up to cubic millimeters per month, right? Which would be about 1,000 times faster than current methods. And most importantly, you get to a point where you could do this mapping for a cost of about 50 cents per cell, which is a similar cost right now to the cost of single cell RNA sequencing, and would really allow you to scale this up and begin applying it to a bunch of different problems, like problems in development, problems looking at like the diversity of different kinds of neurons. So, being able to characterize neurons not just by their transcriptomes, but also by their synaptic phenotype, and problems in disease, looking, for example, the effect of addictive drugs on synapse formation, looking at the effect of neurodegeneration on synapses, all sorts of problems like that. So that's it. I would like to acknowledge a bunch of people, my mentors, Ed, Faye, Evan, people I've collaborated with super closely, Bobe, Andrew, people have sponsored my work, the Hertz Foundation, and the National Science Foundation, the US National Science Foundation. And lastly, just to reiterate, openings for postdocs and PhDs. And if this kind of work, this kind of engineering work is of interest to you, and if you're interested in learning, how to both build these technologies and then deploy them and achieve impact with them, please by all means get in touch. I would love to hear from you. So, thank you.
Well, Sam, this was fantastic. Thank you so much. So we have one question. So if you have a couple of minutes, it would be wonderful if you could answer. So Adam, feel free to ask your question to Sam. Actually, I think you've got 2 questions. Just unmute yourself.
Yeah, I was, okay. Yeah, thank you so much. I don't know whether they can work. Thank you so much for the interesting talk. I was wondering about the specific method for depositing your Slide-seq. I think that the idea was that you uniquely identified each bead using the barcode, so that then anything that bound to that bead had that barcode.
Correct. Yeah.
You could then link like what bound to that bead to the location of the bead, and that's how you could get the spatial.
Correct.
And so you had that. There was that unique identifier of the barcode of each bead. That's
right. So each bead has a barcode on it. When the RNA binds to the bead, you do reverse transcription primed by the oligo that's on the bead, which then has the barcode on it. So then you've transferred the barcode onto this. I mean, like, the cDNA then has both the identity of the RNA on it, the sequence of the RNA, and also the barcode, right? And so you do paired end sequencing. From one end, you get the bead barcode. From the other end, you get the transcript sequence, which you then use the transcript sequence to identify what transcript it is. That's right.
Perfect. Thanks so much. Unfortunately, I'm in the second year of my undergraduate degree, so I'm a little bit far off a PhD. So I was just wondering whether you had any advice for sort of going forward, or whether you had any opportunities as well to get involved.
My advice for people is, my advice for undergrads is always basically, 2 things. First of all, focus on technical skills, because like biological knowledge changes so fast. Like what you really want, like undergrad is the time when you're most able to do things like, learn really hard, like technical skills, right? So like physics, chemistry, machine learning, like all that kind of stuff. It becomes much harder to learn that stuff when you are no longer an undergrad, not because you're like worse at learning, but just because you don't have as much time. So that's the first thing, which is like really focus on the technical skills, and don't worry about like learning neuroscience or biology is like usually my advice to people, because that stuff is easy to learn later. The second piece of advice is like get research experience, period, always. like you have to get research experience. I went to this tiny college that none of you have ever heard of called Haverford College, which is outside Philadelphia, which is also maybe a city that you guys have never heard of. But like the point is that the reason I chose to go to Haverford was because there were no graduate students. The fact there were no graduate students meant that the professors there wanted to do research, and they had to use undergrads for research. So I had like a first author paper by the time I graduated from my undergrad, and that was like super enabling for me. So definitely find research opportunities. It's if you want to pursue a career, in research or whatever, it's very important. And the best way to do that, just reach out to professors. You can reach out to me. I mean, I would be happy to see if I have an opportunity in my lab for undergrads. Don't know, I can't promise anything, but just be aggressive about that. Be really aggressive, because you need to find those opportunities and you need to get them. That will be enabling for you.
Thank you so much.
Of course.
Wonderful. Nikki, you had a question.
Yes, hi, thanks so much for a marvelous lecture. So I've actually got two questions. I was wondering to what extent your university will retain patent rights or ownership rights for technology that's been developed. And number two is, do you have spaces in your lab for current DPhil students to work on summer projects or side projects, of coming to London? Okay.
So answer number two is absolutely get in touch. I would love to discuss stuff like that, projects like that. I mean, I'm setting up my lab, and the lab is not going to be full immediately. And so if you are interested in coming to do, as a visiting student, would love to talk about that. Second of all, answer to your second question about IP. Great question. Yes, the Crick is the Crick's IP policy is similar to, it's like, everyone else's like modern IP policies, which is to say the university owns the IP. You have to take a license out of the university. That's actually like one of the reasons why it's so important to have these relationships with investors, have relationships with entrepreneurs, people who are board members, because what happens is that if you're a professor and you go to the TLO, the Technology Licensing Office, and you say to them, I want to take this technology out, they'll say, great, give us like 20% of the company or something. If you have, like Lux Capital behind you, which is a famous big biotech investor, they invest in a bunch of things. Biotech is one of them, and they have like a billion, 1 point some billion dollar fund size or something. I don't remember, but and like Lux goes to the TLO and says, we're going to put in $30 million, but like, only if you take like 3%, right? Then you have leverage, right? Money is leverage. And so that's part of what I'm doing right now is I'm saying, I don't want to wait until the technologies are done to start thinking about how we're thinking about what the commercial strategy is or what the translational strategy is, right? I mean, if we are going to have to get investors on board or whatever to get things spun out, we want to do that at the beginning so that we know like what kind of data they want to see, blah, 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 right? I mean, fundamentally, what the goal is to invent technologies that are disruptive for biology and medicine, right? Like, if the right path is for them to be commercialized, great. If the right path is for them to be distributed for free, great. But some technologies they have to be commercialized in order to get out. Some have to be given away for free in order to get out. And whichever path is the best is the one that we would follow. But
yeah, thanks so much. Those are great answers to both questions.
Yeah.
Thank you so much, Nikki. And if there are no other questions, I would actually keep up the trend and have two questions. So first would be on kind of the science bit, and as someone who is focused a lot in his PhD on the modeling and on control theory applied to synthetic biology and kind of these complex systems, I would wonder how, for instance, the Slide-seq could be used for how do you integrate this method with like systems biology and perhaps getting more quantitative understanding of the physiological and biological processes. So that would be question number one. And a question number two would be, what really caught my attention in the beginning of your talk was the focus on the commercialization. And I think we had a great question on that from Nikki, but at what stage in your, when you're working on, say, research that would then be spin out, at what stage should you actually start pursuing, at what stage do you know that I have something that can be spun out? What's the right moment? Like, how developed the technology should be? Thanks,
yeah,
thanks so much, Sam.
Yeah, of course. So answer to the question two first is it's never too early. I mean, a lot of academics want to keep their stuff secret, right? I'm really not in favor of keeping stuff secret because, you know, I don't know. I prefer to be working on things that are like, differentiated enough that there aren't that many competitors anyway, right? Like things that are really ahead of what people are doing. And so in that sense, it's never too early because there are two things. Like the first thing is that the problem has to be right. And then the second thing is that the solution has to be deployable, right? And so already when you're thinking about the problem, it's like a good idea to go and talk to people and be like, hey, this is the problem we're thinking about. Like, what do you think about this problem? Are we thinking about the problem the right way, right? And then for example, I had an idea about like a drug screening platform using the Connectomics thing, but I didn't really know, like, is this a good idea? I'm not sure. And I went to Mark Namchuk, who's now at Harvard, who was the global head of R&D at Vertex, which is another big pharmaceutical company. And I told him about it, and he was like, this is a good idea, and here's why. And it was a good idea, but it was a good idea for reasons that were different from the reasons that I thought, right? Like it was a good, so my idea about why it was useful was actually totally wrong, right? But it turns out it is useful. And so then that changes the way that you think about it, changes the way that you pitch it, right? It changes the way that you pitch it to grant funders. It changes the way you pitch it to philanthropists. It changes the way you pitch it to, foundations. And it changes the way that you think about patenting it. It changes the way that you think about what experiments you're going to do, right? And how you're going to write the paper. So that's the answer to the second question. Answer to the first question is like, no one knows what to do with Slide-seq data. And I think it's like a super exciting time if you want to like be working on systems biology to think about that problem, because the fact of the matter is that, the analytical methods that allowed us to get images that look like, that look like this, right? To actually pull out these spatial gene expression patterns. Like I invented them, just like pulled them out, like the statistical methods, I just like came up with them de novo because there was like nothing there, right? For systematically figuring out how to integrate spatial information with biology, because no one has had the data before. So now is the first time we have the data, right? And so, yeah, you have to figure out like, how do you structure the experiments? What's the right way to do the experiment, right? And so I think it's like that is a problem. That is a still open problem. What's the best way to structure? What's the experimental design for spatial experiments is a thing people haven't figured out yet. And then once you have figured that out, how do you do the analysis, right? I mean, I think that the thing that we really learned was that the best thing to do here is like the time series. This story, the story about like looking at the injury, how the injury progresses over time, was much more interesting, actually. And if you read the paper, it's much more interesting than this story, which is like the de novo discovery of these gene patterns, just because this tells you about dynamics, and everything that is interesting in biology is dynamics, right? So
exactly.
Yeah.
Well, yeah, no, that's great, because like that's what we're looking at, the dynamical systems. And I feel often, for instance, you're talking to cancer researchers and they will be like, oh, I've got this huge data set of miRNA expression. And I'm like, how many time points do you have? And like, one? I'm like, okay.
I know. So, and I see there's another question that I can answer just quickly, and then I'm gonna have to go. But about expansion microscopy, this is a question that always comes up. I don't think that there has ever been a talk that has mentioned expansion microscopy that has not had this question. So it's a great question. How precise is it? How on the larger scale? The answer is, hundreds of person hours in Ed Boyden's lab have been dedicated to validating the precision of expansion microscopy. and it's extremely good.
Extremely
So
good.
on the left is, sorry, I didn't let you ask the question, but on the left is actually a 20x expanded, an image of a 20x expanded sample, where like those speckles that you can see there, so these are dendrites, right, that are expressing GFP, or I guess like BFP maybe is the blue one, and those speckles are individual molecules of protein, right? Those are individual like GFP molecules. So, you know, it's expanded 20x, which means the diffraction limit there is like 15 nanometers or something. Why is it so good? It's so good because you digest away all the proteins. You like anchor in the things that you want to look at, and you digest away everything else, right? And so, so the expanding gel is mostly just a gel. It's like mostly empty, right?
Okay.
And so, you know, basically it ends up like the distortions end up being on the size of the, on like the scale of the pore size of the gel. And for the gels that we use, the pore size ends up being like 5 or 10 nanometers. So the actual distortions are like 5 or 10 nanometers.
And I believe you have basically pictures to compare from, I don't know, from STAD and from expansion microscopy from the same slides to show that actually.
Yeah, they did like STORM. You can compare
STORM,
like
okay.
to or like to STED or something. In the original paper, you gotta like every single paper that Ed publishes, he has data about that. So this is like the iterative. The image on the left is from the iterative expansion microscopy paper, which came out in Nature Methods, I think, last year. And they have STORM data. The original expansion microscopy paper, which was Science 2015, had that data. So.
Because I recently encountered this expansion microscope, and I thought to deploy it in my lab because we really didn't know, you know, to kind of separate the protein expression on the presynaptic and the postsynaptic cell in the auditory brainstem. So, and I thought, well, since I don't have any access to STAD or STORM, it's just an extra step. Okay, it was very cool. That's very nice talk. Thank you very much.
Thank you.
Well, Sam, thank you so much. I think this was fascinating, and I think also the discussion was great. So I think everybody should have Sam's email available if you want to get in touch. And I'm really grateful for this wonderful, wonderful introduction, not only into this topic, but also into general thinking. So thank you everybody for joining. Have a nice rest of the day, and we are looking forward to being joined by you in the next events of SynBio.Oxford, Oxford University Synthetic Biology Society. Bye, everyone.
Thanks, guys.